Okay, this is week number 6 of the winter quarter and 599J, the weekly seminar on bioengineering. And last week we had 2 PhD students. Today we have one of our alumni, Dr. David Basiji, who is a founder and chief technology officer of Amnis Corporation in Seattle, which is developing high throughput cell imaging technology for research, drug discovery, and clinical diagnostics. He received his BS degree in biophysics from Berkeley and PhD in bioengineering in 1997 from University of Washington. And currently he is an affiliate assistant professor of bioengineering. And his expertise and areas of interest lie in signal processing. So carefully listen to the areas: signal processing, expert systems, imaging, bioinstrumentation development, and rare cell detection. Broad, which is typical of bioengineering, the education and training. And research focuses on technology development of optical imaging and spectroscopy and their applications in biomedicine, which is today's presentation topic. So David will give us a presentation with the title of High Throughput Multispectral Imaging for Cellular and Molecular Analysis. David?
Thank you. Can everybody hear me?
That's for the remote audience.
Okay. I'm fighting a cold off, so I may pause at points here. There's going to be some dim imagery, relatively dim imagery, so if there's some way to kill the bright lights up in that corner, that would be great. Thanks again to Dr. Kim for inviting me, and as he mentioned, I graduated in '97, so that makes 12 years since I started the program. Yeah, it seems like just yesterday. But after leaving bioengineering, I founded a company with a partner of mine, Bill Orton. That's a long story, an entire lecture, an extremely boring one in that. But we started a company to develop technology for imaging cells. The concept here is that if you look at microscopy and flow cytometry, which are the 2 dominant technologies for cellular analysis, there hasn't been a fundamental architectural redesign of those technologies in quite some time. There's a lot of new advancements, confocal microscopy and so on, but we wanted to take a look with a clean sheet of paper at what you could do with modern laser technology and DSPs and modern imaging. And so we came up with technology that we call ImageStream, and founded this company Amnis to develop it. So Amnis is Latin for stream, and the name ties in with the first application that we sought to apply the technology to, which was looking for rare fetal cells in maternal blood during pregnancy and try and make the amniocentesis obsolete. If you're familiar with amniocentesis, it's a fairly risky invasive procedure to diagnose the presence of birth defects, and it's very expensive and stressful for the parents. And so we sought to turn that into essentially a risk-free blood test, where you just take a little bit of blood from the mother and look for the fetal cells and diagnose the karyotype of the fetal cells. That's a very, very difficult application, and we still think we have the best technology to do it, but we've since gone on to identify other applications that we can go after in the nearer term as we develop the technology towards ultimately being able to do these rare cell detection applications. So a little bit about the company. We were founded in January of '99. We're downtown Seattle, 21 people. A few acknowledgements up front. We have about $1 million in grant funding from the National Cancer Institute, and we currently collaborate with Amgen, the University of Washington, the Fred Hutch, and the British Columbia Cancer Agency. So I mentioned we wanted to take a look at the architecture of cellular analysis technology, and if you look at microscopy, the real strength of microscopy is the amount of information it gets from each individual cell, generally in the form of an image. And that image can either be a transmitted light image, a bright field image, or a scattered light image, so-called dark field, or increasingly these days fluorescence, where you take a probe that's specific to a particular molecule or structure within the cell, you label it with a fluorescence probe and excite it with light to get a report back. And that report is spectrally encoded. The different probes that you put in the cell tend to have different colors, and that's how you identify different structures and molecules within the cell. And so imagery is really essential these days for looking at cellular information. The problem with imagery is that it tends to be slow to acquire, and slow to process. And to deal with high throughput analysis, folks about 30 years ago came up with the technology of flow cytometry. This is where you entrain cells in fluid suspension, flow them through an instrument at high speed, interrogate them with lasers to excite fluorescence, and then measure the intensity of the different fluorescent colors that are emitted from the cell. So these things can run at tens of thousands of cells per second. So you can get very, very good population statistics and find out what's going on if you can encode the readout of your experiments as a particular change in intensity of a particular color. But the information content from any given individual cell is relatively low, 7, 8, 9 intensity measurements. And so we really wanted to try and combine the population power of statistical power of flow cytometry with the information power of microscopy. And so we came up with this ImageStream system, and I've got a diagram of it up on the screen here. So the system actually is generic. It can work with cells in flow, which are shown in flowing in a single file line through a glass cuvette here, or it can work with cells on a microtiter plate or a microscope slide that's in motion. The key here is that the technology is really optimized to work with cells in motion, be that either cells that you've put in motion by driving them with a motor, or that you have in flow and therefore can't stop and therefore can't use conventional imaging. So I have an animation here that illustrates high throughput imaging as it's conventionally done. This is done using a technique called flash on the fly. Okay, so what we have here is once this thing decides to start, maybe it won't. There we go. Okay, so we have a cell in motion. That can be a cell in flow, or it can be a cell on a moving substrate. There's a grid here representing a detector. In our case, it's a CCD detector, but in any case, it's an imaging detector. That is, it has different pixels. There's an imaging system that's not shown here that projects an image of the cell onto the detector, and then there's a light source over to the left, and it's a generic light source. It can either be transmitting through the cell or exciting fluorescence from the cell. And the key to high-speed imagery using what's called frame detectors or detectors that read out an entire two-dimensional image at once is you can only expose that detector to the cell for less than the time it takes that cell to traverse one row of pixels. If you keep that light source on, or if you have a shutter in front of your detector, or you're electronically gating it, the total integration time for signal has to be less than the time it takes that cell to cross one row of pixels. If it's longer, you're going to get streaking in the image, and your image fidelity is going to go down. So you can image at very high speed this way, but you don't get a lot of time to collect signal. And when you're looking at fluorescence, you're generally signal limited, and so you want to increase that integration time. So what we employ is a technique called time delay integration. So in TDI, what you have is the exact same schematic layout here. The difference is in how you read out that detector. So instead of reading out an entire two-dimensional array of pixels and then processing that image, what we do is we read out just the bottom row of data as a one-dimensional pixel vector. And every time you read out the bottom row of data, you take all the rest of the data on the chip and you shift it down by one row. and you repeat this process over and over again. So it's a shift, translate, shift, translate. And what this does is it causes the electronic latent signal on the chip to translate down the chip linearly. And if you know how fast that cell is moving, you can control the readout rate to synchronize the two processes as shown here. And so if you can accomplish that, then it's exactly equivalent to panning a camera to track a moving object. And what this allows you to do is keep your illumination source on for the entire time that cell's in the field of view in front of the detector, as opposed to just the time it's in front of one row of pixels. So basically your integration time goes up proportional to the number of rows in that detector. And we currently use a detector that's 512 rows tall, so we get between 2 and 3 orders of magnitude increase in signal integration time. So that's how we combine high throughput by keeping these cells in motion with high sensitivity. But that sensitivity doesn't do you a whole lot of good unless you can get more information from the cell. And so we take that extra signal and we process it optically to extract more information. We've got a couple different ways to do this. In one case, we put a prism or a grating out here. And so we're collecting in this schematic. We've got our cells in flow. We've got a couple illumination sources here. We've got a laser showing illumination from the side. We've got a bright field source illuminating from behind, microscope objective, and then some intermediate imagery. And what happens out here is we can put a prism, and if we do that, then the light will be dispersed in the horizontal axis. Keep in mind we have a one-dimensional, essentially a vertical line of cells, and we've got a two-dimensional detector. So we've got all this lateral real estate that we can use. And so what we do is we take the image of the cell and we spread it across that real estate to get more information out. If that's a prism that we put in there, then what we're getting is a spectrum of that cell. If we put what we call our spectral decomposition element, what this thing is a fan-shaped array of color-selective mirrors, dichroic mirrors. So each mirror reflects one color band and transmits all the rest. And so by stacking these on top of each other, you can cause, in this diagram, we show violet light coming out to the left, we show red light coming out to the right, and intermediate spectral bands in between. So we're basically imparting an angular shift on the light as a function of the spectral content. And when you refocus that light back down onto the detector, which is shown enlarged here, what this causes is different spectral images of the cell. If the cell is shown here over to the left, then each spectral band is imaged in the different spectral channels, of which we show 5 here. I'm going to show you some 4-channel imagery today. We're currently up to 6 channels as of actually yesterday. And so what we get by doing this spectral decomposition or spectroscopy is a lot of optical image processing done in the optical domain. We don't have to go in with a superimposed image like this that we've grabbed with a camera and try and discriminate the red probe intensity from the blue background from the scatter underneath and so on. It's all spatially segregated from each other by spectral content. So if we're looking at the red probe, we can quantitate it on more or less a black background. So it makes life a lot easier. A couple other things before I leave this slide. We show here a bright field illuminator. If we turn that illuminator on and it's transmitting red light through the cells, then what will show up in the red channel here is a bright field image, a transmitted light image on a gray background. So the areas of high optical absorbance in the cell will show up darker. At the same time, we can take the laser light that's scattered sideways here and image that in its own spectral channel. So that's the equivalent of dark field microscopy. And then the fluorescence that's excited by that laser is imaged in the other spectral channels. So we can perform bright field and dark field and multiple colors of fluorescence imagery simultaneously at high throughput. When I say high throughput, I'm talking typically between 100 and 1,000 cells per second. So again, before I leave this slide, one of the things we do is we have a velocity detection system that looks at it amplitude modulates the signal scattered light from the cells is projected through a grating. So it's kind of like having a car with headlights on at night and looking at it through a picket fence. As it drives by, the headlights are going to flash through the picket fence, and the faster it's going, the faster they're going to flash. So we do a Fourier transform on that signal, find the frequency peak, and that peak is proportional to the velocity of the cells. And we use that as the feedback control for the readout rate of the TDI camera. Another thing that's not shown here is when you're in flow, you have access to all four sides of these cells. And so that allows you to do these multimode illumination, but it also allows you to do stereoscopic three-dimensional imaging. If you take a second TDI imaging leg, just like the first one, and send it out to the side here, both of them can look simultaneously at the cell stream and image independently of each other. So you can essentially get an XYZ coordinate for every feature in that cell, again with no speed or sensitivity penalty. So this is just a quick graphic of some data showing the difference in signal levels between TDI and conventional frame imagery. So this is 3-channel data of chicken erythrocytes. These are nucleated blood cells from chickens that we put a DNA binding dye on, a fluorescent DNA binding dye on, and illuminated from the side with a laser to excite that fluorescence, and from behind with red light. So we're showing in this case red on the right. This is a bright field image of the cell. And then over to the left is a fluorescence image of the nucleus. If you then, this was done on an early prototype. We have everything on magnetic bases, so you can pop cameras in and out and so on. We popped a frame camera in, set the exposure time to the same exposure as the TDI camera, and with the same sample. And what you see is in the case of bright field imagery, you get the images streaked out, in this case over 96 rows, because the height of the TDI camera we were using was 96 rows in this case. So for the same exposure time, it streaks out over 96. You can still collect fluorescence imagery, and you can add up all that signal, but because the same signal level is now spread over 96 times as many pixels, each of those pixels contributes its own noise to the detection, and so your signal-noise ratio goes down by about a factor of 10 there. You can cut the exposure time by about a factor of 96 in this case, and do clear frame imagery, which you may be able to see in this dim image. But so there's a little bit of fluorescence light there, and there's a bright field image here. But of course, the signal is 96 times dimmer, so you sacrifice a lot. So in terms of the data characteristics, what we have here is 4-channel data of an engineered sample. These are polystyrene beads that have been labeled with one or the other of 2 different fluorescent dyes, or no dye at all. And what we're showing here is simultaneous bright field, dark field, and the 2 channels of fluorescence imagery. So each bead came down the pipe and was imaged with the TDI detector. Each bead transited all, in this case, 96 stages of the TDI detector, and was read out at the end. And what we do is we have a real-time algorithm that's monitoring every pixel that comes off that chip. And it does a histogram of the background noise characteristics of that chip. So it can analyze the intensity of a particular pixel, and if that pixel is statistically unlikely to be background noise, it decides that belongs to imagery of interest, and so it segments it out of the raw data stream. And so that way we can cut out all the dead space between the cells as they come down. I should mention we're producing imagery at about 60 megabytes a second off this detector, and so the data flow is quite high. You have to have these upfront data reduction schemes to make it feasible to use affordable hardware. So all this is done with Pentium PCs and so on. So anyway, back to the data. So here we can focus on this one bead. What we're showing here is an absorbed light image in bright field. At the same time, you can see the scattered light off the front and the back edge of the bead. In this case, we have the laser light coming in from the left side. And then that laser light was exciting fluorescence. In this case, there was no fluorescence because we don't have any imagery in either channel. But in the fluorescing channel, this bead produces an image, and in the PE channel, these beads produce an image. And so you can see it's very simple to, at a glance, tell the characteristics of these beads. And as I'll show you going forward, there's very complementary information in the different channels. You can do everything a flow cytometer does with this data. You can go through and add up the total intensity in that bead in dark field or optical density in bright field or in the fluorescence channels. Here we're showing total intensity of the fluorescein signal versus the phycoerythrin signal. You can see there's a population of unstained beads, population of FITSI beads, and a population of PE beads. And in our system, of course, every one of these dots, which represents an individual bead, is correlated to an image set, and so we can go click on those dots, as I'll show you, and bring up the imagery. A little more interesting, this is cellular data. So let me draw your attention to the right here. These are K562 cells. This is a hematopoietic cell line. And we've labeled these cells with a DNA binding dye from molecular probes to get nuclear morphology. As you can see here, we've got a dual lobe nucleus. We also put a monoclonal antibody against the transferrin receptor on these cells and labeled it with the Cy3 dye. And so you can see the distribution and intensity of the cell surface receptor, and then again bright field and dark field imagery. You can see that there's complementary information here. The areas of high scatter don't necessarily correlate to areas of high absorption, and vice versa. And so you get a lot of different forms of imagery here. Looking on the left, what you're seeing here is actually the raw data as it comes out of the detector. And after that real-time segmentation or object detection, there's a fair amount of background signal here. The bright field leaks into the other spectral channels, but because the system's deterministic, we know what the filter characteristics are, we know what the dye emission spectra are. We can go through and correct all that crosstalk, put it back in the channel that it belongs. So this is just backing up a step. The ImageStream is really the first system to do fluorescence imagery in flow. People have done imaging using bright field primarily in flow with intensified cameras and very bright pulse sources. But when you're doing fluorescence, you need to be able to probe these cells. And with all the probes that have come along in the last 10 years or so, genetic probes, protein probes, and so on. And so what we had to do was redevelop all the probing protocols that have been developed in microscopy for labeling specific genetic sequences, labeling proteins in the cytoplasm, labeling molecules on the cell surface. And the problem is that when you're using cells on slides, you literally have a handle on those cells. You can pick that slide up, you can dunk it into different wash baths, reagent baths, and so your probing protocols are relatively simple. In our case, because we're in suspension, we don't have that luxury. We can't just grab onto those cells and manipulate them. And so we had to do a lot of suspension-based probing. Protocol development. And so what we're showing here, these are fluorescence microscope images that we used a fluorescence microscope before we had a functioning prototype to develop these protocols. What we're showing here is an intact cell labeled with 2 chromosome-specific probes, in this case against the Y chromosome. So you can see this is a female, X chromosome. This is a female cell. Here we're using X and Y. So it's a male cell. We can also look at specific gene loci. Here we have HER2/neu, which is amplified in roughly 30% of breast cancer cases. BCR-ABL, which is a translocation in the Philadelphia chromosome present in leukemias. But we wanted to take it one step further and be able to combine the genomic probing with the probing of the other cellular compartments. And so this is some work done by Linda Finch, the first biologist that we brought on board. She's brilliant in the lab, and she was able to come up with these protocols in very short order. Here we're showing a red fluorescent probe again against the X chromosome, combined with a cell surface probe. Here we have X chromosome probe with monoclonal antibody against fetal hemoglobin, so we're doing a cytoplasmic protein probe along with the genomic probes. And then here we do all three compartments at once, cell surface receptor, the fetal hemoglobin, and the chromosomal probe. So again, along the lines of getting more information from the cells, it's attacking it not just from the instrumentation angle, but also from the probing angle. So here's some very early data. I apologize for the quality of it. This is 2-channel data, bright field and 1-channel fluorescence. These are peripheral blood mononuclear cells, essentially white blood cells that have been probed with an X chromosome probe. And you can see the bright field morphology along with the nuclear morphology from the background stain, and then the presence of individual fish spots corresponding to the individual chromosomes. We've also done this with cancer cells. It's a bladder cancer cell line that exhibits chromosome 9 trisomy. We put a chromosome 9 probe in there, and you see 3 fish spots in a large number of these cells. If you've ever looked at fish spots through a microscope, typically you do it with a very high numeric aperture oil immersion lens. And when you do that, you have very little depth of field, and so you're constantly racking focus up and down to find these fish spots in any given cell. If you want to do this at high throughput, these cells, you only get one shot at imaging these cells as they pass through, so it's tough to rack focus. So what we do instead is we sacrifice some sensitivity. We use the sensitivity of the system and sacrifice it, trade it off for depth of field. And so what we do is we take we put an iris in the back aperture of the system, and we close that iris down. So we effectively cut the numeric aperture of the system. We can afford to do that because we have excess sensitivity. But by doing so, you get a squared function increase in the depth of field. And so you get a very large fraction of these cells with in-focus fish spots, even though they've only made a single pass across the system. So it's just another way to translate one instrument characteristic into another characteristic that specific for a particular assay. How am I doing on time here? Pretty good. So this is just a placeholder to give a little demo of the application software. So when you're processing cells at hundreds or thousands per second, you've got just a nightmare of data to deal with. We regularly deal with image files that are 250 megabytes in size. And so we had to come up with software that would facilitate both the inspection of individual cell images to get the most information and the inspection of large populations. And so I have to apologize again for the quality of the cell imagery. This was taken on a prototype that with about 1/4 the resolution that we're running with now. But just to give you a little flavor for what the software does, we have an image gallery over to the left here. And so we can show every cell is given a unique number. In this case, it's imaged in 4 channels, bright field, dark field, and 2 fluorescence channels. We pulse colored those channels so that you can get a sense of what they would look like through the microscope. We can actually go through and superimpose them so it even looks more like what you'd see through a microscope, but sometimes it's easier to analyze the cells looking at them decomposed like this. So you can go through and you can select individual cell images and get all the parameters that are calculated on each cell in the upper right here. And so we have up to 6 channels of spectral imagery, and we calculate between 15 and 20 features on each image of each cell, each channel image of each cell. And so that means you've got over 100 parameters to work with. And so when you click on each of these cells, all the parameters that you're interested in can be displayed over here. You can also look at statistics of the population as a whole, the bright field area, CVs, means, max, mins, and so on. You can select any of the parameters that you want, or all of them. And you can go through and you can inspect your imagery and select individual cell images or image sets that are of interest to you and create a population that way. Or you can go through and do bivariate plots, as is shown here, of different parameters against each other. So here we're showing the intensity of the annexin stain in this apoptosis assay. The annexin stains, it binds to phosphatidylserine on the surface of the cells, which is generally only present on the outside of the cells if they're undergoing apoptosis. And so in apoptotic cells, you get a nice green signal. And so we're plotting total intensity of an exon versus a nuclear stain 7A AD. The nucleus in apoptotic cells tends to bunch up and fragment, and so imagery is really a good way to analyze apoptosis. And what we're showing here is a population of about 500 cells that we've plotted here. You can go and click on any one of these dots. Again, they correspond to cell imagery, and you can go and inspect any image that you want. I've gone ahead and selected images of interest, and each of those images is highlighted in red in the population here. And so if you go and pull down image sets, you can find out where they fall in all your graphs, and they're displayed in all the graphs simultaneously. So it's again, the concept is to facilitate the researcher's ability to really explore their data. I'll try and keep the commercial pitch to a minimum here, but this is the latest prototype we have at Amnis. It's an alpha prototype. It fits in a box about the size of a flow cytometer, and sucks samples out the front, runs them through, generates the imagery, and we currently use 2 computers to run the system. We'll shrink that down to 1 by the time this thing's released. So in terms of what this thing does now, what we're doing is we're performing multimode imagery, simultaneous bright field, dark field, and multiple channels of fluorescence in up to 6 channels simultaneously. Right now we have about a half micron pixel size, where the optical system has about a 0.75 NA, so it's capable of about a 1/3 micron resolution. By the Nyquist criterion, we should be running at about a 0.15 micron pixel to fully take advantage of the optical resolution, but it's a trade-off of speed for resolution, and so we set the initial system at a half micron. We'll probably be bringing that down to about a 1/4 micron in the near future here. In terms of sensitivity, the way people in the flow cytometry field quantitate sensitivity is in terms of molecules of equivalent soluble fluorescein. So if you have fluorescein dye on a sample, how many molecules is your detection limit? We're currently running in the few thousand molecule detection limit, which is slightly worse than typical flow cytometer. What this means practically is that if you can see it with your eye in a microscope, you can probably see it with the ImageStream. And we have high sensitivity variants of this system in the lab that can get down into a few tens of molecules of equivalent soluble fluorescein, and so on a per pixel basis. So we believe we can get actually down to the single molecule detection limit at high throughput. Once you're here, you're talking having to integrate on a microscope for several minutes to be able to get detection. The system quantitates at 10 bits per pixel off the camera, and at about 300 pixels per image. That means that you've got roughly another 8 bits, assuming your signal is uniformly distributed over the whole cell, and you're imaging about 300 pixels per cell. You get about 18 bits of theoretical intensity resolution over the cell. If that signal is concentrated in fewer pixels, then your intensity resolution gets closer and closer to 10 bits, which is about 1 part in 1,000. So for typical flow cytometric assays, you're looking at trying to achieve about a 1% repeatability. So this is sufficient for pretty much anything that's done in flow cytometry right now. Throughput at this resolution is about 100 cells a second. We can bin our detector and give up resolution for increased throughput. Talked about the crosstalk compensation and the probing protocols. And you saw a bit of the image analysis. One of the things we're going for is a completely self-aligning, self-diagnostic system. So the sophistication in the instrumentation is really designed to make it easy to use and reliable. And we want to make as few knobs available to the user as possible, because if we put them in there, they're going to get turned and the system's going to get screwed up. So we try and keep the system as automated as possible. So the last few slides have dealt with where we are now. In terms of where we're going, one of the goals is because we're doing multispectral imaging, for every location in that cell, we're getting an independent measurement of 3, 4, 5, 6 different fluorescent probes at that location in different spectral bands. And so what this means is that We have the potential to do probe multiplexing. That is, where you take instead of having an individual color per fluorescent probe, as shown in this cartoon here, if you can do the multispectral imaging, you can potentially assign a unique color code to each probe. And if you do that, and these probes are spatially distinguishable in the image, then, for instance, you can tell the white probe, shown here as having three primary colors, blue, green, and red, and you can distinguish that from the magenta probe, which has blue and red, but no green. You just go to the corresponding location in each channel and look for a signal. And if you do that, then the number of probes that you can theoretically use in a cell goes up as a power function of the number of colors. So in a three-color system shown here, you can do 7 different probes, up to 7 different probes. You can't see all 8 of them because there's a black probe and it's not emitting any light. So it's not strictly a power of 2. But if you go up to 5 fluorescent colors, you're talking 31 probes, 6 fluorescent colors, 63 probes. So, as opposed to fluorescent microscopy, where you have typically 3 fluorescent colors, or flow cytometry, where you can get 6, 7, 8, even 10 in certain research instruments, the number of probes you can do in those instruments is generally proportional to the number of colors that you can measure. In this case, we can get that up to an exponent. I talked a bit about ultrasensitivity. This is some very, very early feasibility data that we took. This is an image of a bead that normally falls at the detection limit of a flow cytometer. In our system with this intensified TDI camera, essentially every pixel in that image has the sensitivity of a photomultiplier tube. And so because the noise that you're throwing into that pixel is just corresponds to that little half micron by half micron space in the object space, you're actually excluding an awful lot of background signal. And so you can get very, very good signal-noise ratio in a pixelated image, even if you're just looking at total intensity. And so with this early prototype, we were able to demonstrate 60 molecules per pixel detection limit. We don't think it'll take heroics to get down to a single molecule, as I mentioned. I'm running out of time, so I'm not going to spend a whole lot of time on the molecular analysis side of things, but I will talk about it briefly. So we've been talking about cellular analysis until now, but we can also apply the system to molecular analysis in a very analogous fashion to microarrays. So if you're familiar with these, you can put DNA spots down on a two-dimensional surface. It's spatially encoded. You know the XY coordinates of each sequence. So when you hybridize an unknown DNA sample to it, you can look at where the spots light up and then decode what the sequence of the probe must have been. If you want to get to very, very large levels of libraries, that is, very large number of spots, you run into a problem where you run out of real estate. Your array gets very large, and you've got this problem where you've got molecules in solution that are trying, in a three-dimensional diffusion space, trying to hybridize to molecules that are on a two-dimensional surface, and so the kinetics start to work against you as that array gets bigger and bigger. And so if you are trying to get to very high levels of molecular analysis, analyzing lots of different molecules simultaneously, and you want to get into the millions or even billions of different molecular species, you really want to use a three-dimensional structure to hybridize those molecules. So there's been some development along these lines using bead technology, where people put different molecules on optically encoded beads, and then hybridize their unknown to this bead library, and then based on which beads light up, you're getting the equivalent of a spot on a microarray. The problem is that it's difficult to generate very large diversity in these bead libraries. You're trying to generate millions or billions of unique optical signatures that you can then use to discriminate what molecule is on the surface of that bead. So there's a couple ways to do this. People currently do it by optically encoding, intensity coding beads. That is, they put a different ratio of usually 2, sometimes 3 different dyes on these beads. And then the ratio of the 2 different dye colors and their intensities gives you the optical signature for that bead. And you can analyze that kind of thing in our system by putting a prism in there and doing essentially flow spectroscopy, as we've done here. So what we're showing is a bead. You're looking at scattered 488 nanometer laser light here. So this is essentially a dark field image. And then out to the right is the emission of the fluorescent probe that's attached to that bead. And in this case, we're looking at a 3-color example. So this is a red bead, this is an orange bead, this is a yellow bead. This one's got both colors. You can, if you do an intensity profile of these beads, you can find the peak positions, you can integrate onto the curve, get the intensities, and you can decode those beads. But what we really want to do is leverage the multispectral imaging to increase the number of beads in the library. And the way we plan to do that is by essentially taking a large bead that your molecule of interest is adhering to, and encoding that bead with little fluorescent reporter beads on its surface. And in essence, every one of these reporter beads is analogous to one of these intensity-coded beads that I just talked about. But you can have several different species, many different species, on the surface of this carrier bead at the same time. And so the way the encoding goes is you image that bead in the ImageStream system, and you look for the optical signature. So in this case, we're showing a reporter bead that has a yellow and green fluorescent emission, and we can distinguish that, just like we did with the multiplexed probing, from another bead that has blue, green, yellow, and red, and that's distinguishable from green and red, which is distinguishable from a bead that just has green and blue. If you do that, jumping ahead here, what you do is you take this intensity coding combinatoric formula, shown here, for example, 2 colors in 10 intensities. There's a company called Luminex that's doing this. You can produce 100 different unique beads with that kind of system. If you do this reporter labeled bead scheme, that formula gets raised to another exponent, and that exponent is basically the number of different kinds of reporters you can put on the surface of that carrier bead. So this kind of combinatorics, because you've got an exponent on an exponent, just explodes. And with a very reasonable number of colors and a very reasonable number of intensities, you can generate optical diversity in the billions. So now, essentially, the limitation on your ability to analyze a complex molecular mixture is limited only by your ability to read these beads fast enough. And so that's an area that we're going to be pushing forward in the future here with the ImageStream system. Then you can do things like sequencing by hybridization of DNA stretches by looking at essentially the hybridization pattern of different beads. Here we show a bead library containing every possible DNA temer on the surface of the bead library, and so if you have a stretch of unknown DNA, you're going to get a hybridization pattern in tenfold redundancy, and that'll allow you to not only sequence a stretch but identify changes, in this case a SNP at a particular location. You can profile people's immune systems, look at what kind of antibodies their immune systems produced, and so on. So in terms of going forward, we're developing a number of multi-parameter image-based assays for cells. We're pushing forward on the sensitivity front and the 3D stereo imaging. We're working on very high throughput systems that get from the 100 to 1,000 cells per second into the 10,000 cell per second range. Again, this is sort of worst case scenario, using a single filed row of beads. We can also image beads side by side and get a multiplier on that. We ultimately want to have real-time morphology analysis. So as these cells come down the pipe and are imaged, we want to be able to analyze that image on the fly and decide whether or not we want to sort that cell, and then put that cell in a droplet of fluid and electrostatically sort it, just like a flow cytometer does. To be able to do that, we need very, very high-powered computers running at real time, in hard real time. So that's for the future. Right now, as I said, we're using off-the-shelf hardware. But in addition, we're looking at this multiplexing of both cellular probes and large bead libraries. Again, it comes under the heading of enhanced signal processing, so requiring greater and greater sophistication on the hardware and the software. We're also looking at microtiter plate, microscope slide, and microfluidic versions of the technology. And ultimately, we'll want to get the system out for not just research, which is going to be our first product, but for use in pharmaceutical discovery and development, and ultimately in the clinic. So just to wrap up, I'd like to acknowledge not only the Amnis staff, but our collaborators here at the UW at the Cell Systems Initiative. Bob Franz has been a great supporter of ours, and he'll be getting one of the first beta systems to come out of Amnis. We've also been working with Leon Wheelis, Professor Emeritus at the University of Rochester, and Dr. Reeder. Those bladder samples came from Dr. Wheelis's lab. Peter Rabinovich and Dr. Vasella here at the UW. And then over at the Fred Hutch, Andrew Berger, Michelle Black, and Christina Pinson. And once again, I'd like to thank the NIH and the NCI for their support. So that pretty much does it.
Question there.
How do you do tumbling of either the beads or the cells as they go down your field?
That was actually the very first question we had to ask ourselves. One of the first things we did before we built any hardware was develop a Monte Carlo simulation of the technology. And so we had virtual cells running in flow, with virtual misalignment and speed errors and so on, and all the theories said it should work fine. And if you calculate for a 10 micron diameter cell, worst case tumbling is a pixel out at the periphery would blur from one pixel location to the next. The tumbling rate has to be extremely fast, and there's two things working in our favor. One is that even though we're integrating for 1,000 times longer with the system than you do in a flow cytometer, it's still only a few milliseconds. So on those time scales, the viscosity just doesn't allow it to happen. And we've run sperm through the system, which is a very good test for tumbling because it's got that long flexible tail. And when it's sticking out to the side, you've got worst case shear on that cell because the fluid in the pipe has a parabolic velocity profile. So when you get that tail sticking out to the side, it really wants to bend. And you'll see degradation in the contrast out near the tip if you've got any tumbling going on. But all of our measurements say that it's completely under control. That the Reynolds numbers here are 0.004 thereabouts. I mean, it's just incredibly laminar. It's tough to get turbulent flow, much less chaotic flow in this system.
Question?
I think you may have just answered my question. That is, this method depends on being able to determine the velocity of the cell. And the question was whether you're determining individual velocities of cells or just the fluid flow in general.
Well, because we're taking a scatter signal off the objects, we're measuring the velocity of the objects themselves. But we do measure it over a time interval of roughly 1/10 of a second. So what it's doing is it's acquiring a time series of signals from the scatter detector, and so what those signals look like is a big Gaussian envelope corresponding to our laser illumination, and then within that is the amplitude modulation due to the grating. And we typically are looking at 5-10 objects in a single time series. So when we Fourier transform that, we're getting contributions from multiple cells at the same time. So we're calculating an average velocity and updating it at about 10 hertz. So it's compensating for slow drift in the system, changes in fluid velocity and so on. It's not doing a cell-by-cell analysis. We have run it in cell-by-cell mode. Basically, you need to reduce the concentration of the cells so that on average you have only one cell in the field of view at a given time, and that works fine as well. And that's the strategy we'll use for non-hydrodynamic focused flow. So in a microfluidic chip, for instance, where you've got cells that can appear anywhere in that cross section of that channel, they can be at radically different speeds from cell to cell. And in that case, you want to do a cell-by-cell velocity detection. You want to do it upstream of your imaging system so that you can adapt before it gets into the field of view. In our system, the two fields of view are coincident, but because we're hydrodynamically focused to basically the center 10 microns of a 250 micron velocity profile, we get almost no cell-to-cell velocity variation. I mean, they're basically all coming down. It's only slow drift that we have to worry about.
Sayan?
With the signal coming out into different spectral bands, can you comment on the kind of distortions you? Excuse me.
Can you repeat the question?
Yeah, I'm sorry. So the question was, because we're doing this spectral decomposition, what kind of distortion do we suffer as a result? So there's a couple things that happen there. One is we're imaging through multiple filters, and so we can get wavefront distortion by doing that. We need to use very flat filters to do that, and we're actually going towards a monolithic structure of cemented filters. So you can get wavefront distortion. You also get pyramid error. You may have those filters may not be at exactly the angle you expect them to be in the lateral plane, in which case you get a lateral shift of your channel. And from filter to filter, you can get pyramid error, where one image may be slightly higher or lower than the other, and so you've got this two-dimensional variability. So we're very careful about alignment of our system, but after the fact, what we do is we go through and we analyze, in this image file, we've got thousands and thousands of cells, so we can do a correlation from channel to channel and get very, very good subpixel spatial registration. And then once you've accomplished that, the spectral correction, which is sort of the third problem that you have, is easy to deal with, because you've got spectral crosstalk due to all these filters.
Any kind of advice to the graduate students who are in school now? What should they focus on, or what should they learn while in school?
Well, I can tell you one thing, which is that one of the as you brought up at the beginning of the lecture, one of the great strengths of bioengineering is that you have a very broad base of expertise, lots of exposure. The downside of that is that if you're not careful, you can become too generalized and not have the tools you need in any one particular area. So, for instance, what we sometimes see are applicants who have a very broad base of experience, but for instance, don't know how to use the CAD tools that are commonly in use, AutoCAD or SolidWorks or what have you. If we're talking about a mechanical engineering position, or the people who are coming in on the electronics side, are not fully up to date on the image, the capture systems for board layout and that sort of thing. They haven't learned all the tools that you use as an electrical engineer in that case that will serve you in the commercial world. And so and the same is true in optics. There's all these optical design systems, software predominantly that you need to be facile with. And if you're just doing a superficial jack of all trades education, you never really dive in and get the experience programming or using ZMax or using SolidWorks or using OrCAD Capture or whatever the software tool may be. And so that's really the great weakness that you have to watch out for, is make sure that you have the tools in at least one of your areas that are going to be expected of you if you go into the commercial world. Experience running coding in C on a Linux box is great, and our algorithm guys use that every day. But I know from personal experience that's what I did to develop the image analysis software that I did as a grad student. And when I got out into the real world, I didn't know how to use any of the higher level packages that people commonly use for image analysis. And so I had to go back and do a little remedial work to pick up the tools I needed. And so that would probably be my
okay. Final question?
I was just sort of curious what the distribution of your 21 employees was. Sort of what type of people work for you?
So the question is how do our 21 employees break down? There's overhead, which includes me and Bill Orton and our CEO and so on. There's 4 folks that fall into that category. Our biggest group is software and algorithms. We have 6 people there. We have right now a 4-person bio team. That's probably the one that's going to grow the fastest at this point. We up until recently we didn't have any tools for them to work with in terms of ImageStream systems, but now that we're building in-house systems, we can start ramping them up. So there's 4 folks there, and then we also have the balancer in mechanical and electrical engineering.
Okay, thank you very much, David.
Thank you.